Thank you very much, Belle. It's a pleasure and an honor to be here. Belle and I have been on conference calls every 2 weeks for the last year, and so we've gotten to know each other very well. And I've had the pleasure to work with her in the past and for another conference, which one we did, a smaller one we did in Toronto a few years ago. Anyway, so I'm tasked today to get you excited about bioinformatics. And the way I'm going to do that is I'm going to tell you about my journey through my life and how I ended up being a bioinformatician. And it will be, it will have many sort of side stories that I'll be happy to share with you. First of all, and this is something I do in front of most, if not all of my talks, is I put a Creative Commons license, which means that it's a CC BY and CC SA share alike. So that means that you're able to use my slides, and they'll be available after the talk. You're able to use my slides. You can use the one slide you want in your talk. And the sort of infectious part of it is that share alike. That means that if you use one of my slides, then your slide deck has to become shared too. So if you borrow mine, then you have to share forward. And so you can, and remixing means you can take a slide and crop it and use it in your deck and so forth. So this is my email address. It's the hashtags for the conference. And for this session here is as such, and those are my various handles on different social media. So the outline for today's presentation is I'm going to sort of ask you about what you think is, what it is to do bioinformatics. Then I'm going to show you a brief history and then some lessons learned along the way. And I'm going to use the term bioinformatics. Some people prefer to use the term computational biology. There have been many debates, many articles written about the use of one versus the other. And I've had publishers, editors of books tell me, no, you cannot use computational biology. We're going to market better if you call it bioinformatics, and vice versa. And some purists say computational biology is a more pure aspect of it and so forth. I do not engage in those discussions, and I'm going to be using bioinformatics mostly throughout this talk today. And so, so what do biologists do? And so biologists make observations, they make hypotheses, they test them, they challenge them, they conclude things, and then they write about it. In computational biology, or in bioinformatics, that's what we do as well. And so be it an RNA-seq experiment where we analyze all the RNA from a single cell or a group of cells or a tissue, or do a protein analysis type experiments where we sort of do very types. It's another way of doing biology, of doing experimentation. It's another way of doing science. And so as all of you know, I'm sure, the central dogma of biology is DNA makes RNA makes protein. And then I sort of updated it with, then you write a paper about it so that you can actually explain what it is that you saw in this DNA, RNA, and protein sort of effect, and what it does to the cell and so forth. So I'm going to do a little experiment now. I'm going to ask you to pair up with somebody next to you and together tell me what you think. Write in a piece of paper you don't have, but in your phone or something, a few words to describe what bioinformatics is. And then I'm going to give you 1 minute to do this right now. And then I'm going to ask you for what you've come up with your neighbor in this small conference hall. And for the people online, you're following them?
Yes.
Okay, if we'll get some answers from them too. So I'll give you another 30 seconds. So what is bioinformatics? Or what is computational biology? And if you're sitting by yourself, then it's an opportunity to go sit with somebody else. Okay, I'm going to call it a time. So does anybody online have an answer?
So I think Apurva has an answer.
Yeah.
So Apurva says, I think that bioinformatics is analyzing the data generated from the experiments run in a lab that involve DNA, RNA, and protein extraction and amplification.
That's one. Yes. Anybody else in a room? You get
simple one saying trying to understand or ask a biological question using computer tools.
Yeah, that's a very good one. You guys have a? Did you come up with something?
So he said,
Oh, by the way.
Oh, you need microphones? Yeah, you need people to. I'm just going to do one more, so maybe you don't need a microphone.
okay.
Yeah, I'll repeat it. Tell me what you said.
Use computational methods to solve complex biological.
So to use computational methods to solve complex biological things. Yeah, it doesn't. So those are all very good answers, and I would ask 20 other people, and we get 20 different answers, and they would all probably be correct. Here's my sort of answer, which is very similar to what you guys have come up with, which is basically it's the integration of biological themes together with the help of computer tools and biological databases in gaining new knowledge about a system that's in study. So that's my sort of aged, sort of experienced answer. And so it's really, to do bioinformatics, you can write your own tools, but you can also use somebody else's tools. And you can use somebody else's database, or you can make your own database. And so there's all sorts of aspects of doing computational biology. So why do we have bioinformatics? So because we actually have open data. So one of the reasons that has been sort of underlying, sort of unspoken almost at the beginning, was that the fact that there's lots of open data, and because there's lots of open data, then we all need to work on this data, and we all need to build tools to be able to interpret things. So how many of you have ever heard of the program BLAST? BLAST, yeah. Okay, so it's a basic local alignment search tool. So it allows you to search databases for similar sequences of DNA or RNA or proteins. And you can compare nucleotides against nucleotides, or nucleotides against, or translated nucleotides against proteins, or protein against protein, and so forth. So there's all sorts of possibility with this program. And often what we do is, once we get a really strong match in BLAST, we say, well, this protein from this organism is very similar to this protein in that organism. Maybe they have the same function. And so that's what we're trying to project, the knowledge from one organism to another using things like BLAST. So BLAST would not have been invented if GenBank or Atlas, which is another protein database, an older one, which has been replaced by another one now, didn't exist. And so the fact that there's these public nucleotide databases allowed for things like BLAST to be invented. So all of you know, I'm sure, the scientific method, which is basically we make observations, we research a topic area, we make hypotheses, we test with experiments, we analyze data, we report conclusions, which generates more observations and more questions. And so when you do a bioinformatics experiment, you're doing the same thing. You've got sequence, you do a blast search, and then you get an alignment, right? So you have to know your reagents. What's the sequence I'm using to do the search with? Which database am I searching? You have to understand the method. Are you doing a protein-protein similarity, nucleotide-protein, protein-nucleotide, and so forth? So you have to know the methods. And then you have to interpret. So you have to know the reagents, you have to know the methods, and you have to do your controls. So an example of a control is that you would expect in a given blast search, maybe there's some proteins you don't expect to see. And if you don't see them, that's good. But some you do expect to see. And so if you don't see those, but you expect them, then your control, that's an example of a control you could have done in your bioinformatics experiment. So I'm going to give you a brief history of a bioinformatician. So I was born in Quebec City. In my early years, I was moved to Montreal, Winnipeg, Ottawa. I moved a lot as a kid, and I was always interested in biology. So I was always a biologist. I went to high school just up the street here on Polyvalent Lucien Pagé, which is sort of Jarry and Saint Laurent, so it's just north of here. And actually, this high school looks like a jail, but it wasn't a jail. It was actually a nice high school. But we were the first ones into that school. It was a modern, very modern new school. It was big. And so I was listening to Beau Dommage and Harmonium back then. So just to set context. And I had great chemistry, physics, and biology professors. And so I think that was definitely, and I started to read some things. So when I was 13, one of my uncles gave me the double helix to read. And I thought, that's an interesting story. And it was in English too. So now I was a French kid. I spoke some English at the time, but I managed through that one. And another book my mom also gave me at Christmas one year was around then, was Jean Rostand, which he basically wrote, he's a great writer of science, of biology, but he was a loner biology. He would do experiments on his own, and he was independently wealthy from money that he got because his father wrote a very famous novel, and so he had money throughout his whole life. But he worked alone, and that's definitely something that you don't do anymore, but there's definitely many people were doing back then. And so after high school, most people go to CEGEP, and I was in Quebec and I should have gone to CEGEP. I actually skipped CEGEP because my dad got a job in Paris, and so I went to Paris instead, going to CEGEP. And I ended up skipping a year without mixing the school systems and whatnot. And so I came after that year in Paris, then I came back, I went to university. And so in Paris, I actually met Fernand Séguin, who was a very famous, a bit like Jean Rostand, sort of, he was a vulgar, he was like the French David Suzuki equivalent and he was one of the first ones to do this in television. And so UNESCO got the Kalinga Prize, and my father was working at UNESCO, and so we had Fernand Séguin come over for dinner, and I was very excited. Anyway, so after that, I went to McGill, did cell and molecular biology. I was still the French kid, but I read the double helix again. Now I'm 18, so I'm sort of getting more out of it, understanding more the subtle nuances in the book. I did, so like I said, I did biology, and I did not take any computer science courses while I was in undergrad. And I did some biostats. After that, I went to Calgary, and there I went and worked. So I had done cell and molecular and went to a plant sort of biochemistry type lab, and we worked on various enzymes and biochemistry and work on lactuca sativa. Does anybody know what that is? Indeed, very good. Yeah, so lettuce. So it's those are lettuce seeds, and they have some very famous biological phenomena where we shine far red light on them. They germinate, and so they use the reserves in their cell wall. And my thesis topic was beta monocyte monohydrolase in the mobilization of endosperm cell wall in Lactuca sativa CV Grand Rapids. So nothing, I hadn't looked at that title since then. And but during my graduate school, the beginning of my graduate school, I definitely developed my scientific method more, and also further love for genetics. It turns out that I'd done a lot of genetics as an undergrad, and one of the genetics instructors saw my student application when I came in, and she kidnapped me to teach her genetics, to be a TA in her genetics course. I learned how to push flies and do fly genetics in Calgary while I was in the plant lab. So after that, I went to McGill, and actually my supervisor moved from McGill to SFU to the West Coast afterwards. So I went to McGill. So I was in Calgary, McGill, and then back west again. And there I worked on sea urchins, Strongylus introtus purpuratus, and looking at sea urchin early development. And what happens in early development is that there's a class of messenger RNA for the histones, which are involved in the nucleosomes, the structure that the chromatin forms. And there's a early class of histones, and then there's a later class. And then I was looking at the degradation of the early class, so finding out what sequences, so very reductionist, sort of trying to find the elements within the RNA that are responsible for the stability of that messenger RNA. And so during those years, I learned to sequence DNA. I was learning to use software tools to analyze my sequenced DNA. I became sort of the lab computer guy that many labs still have today. And in the lab, we had an IBM PC that a 10 meg hard drive and two 360K floppies. And so I read the double helix again, and I also read Le Hasard et la Nécessité, the Chance and Necessity from Jacques Monod. Jacques Monod had won the Nobel Prize in '65, and he did the lac operon, which is a sort of regulatory network of genes that are controlled together in bacteria. And he also postulated the existence of messenger RNA. So he was quite a smart guy, and he's a very, I would say this book is very much a reductionist book. And so some people fault me with that. I'm too much of a reductionist. I'm not enough of a systems person. But it's because of Jacques Monod. And so it's, he says, if it's true in bacteria, it's true in elephants. And so it was from that point of view, it's not very reductionist. So the biology department purchased a computer, and I was using FastN and FastP, which are predecessors of BLAST. And I moved from McGill to SFU. I learned Unix on the SFU mainframe, and after 7 years, I actually withdrew from the PhD program. So I actually never finished my PhD. But then I got a job at McGill, and I was working on the yeast genome project. And it was a job for a postdoc, but I wasn't a postdoc. But they hired me anyways because I had the skills, the computer skills and the sequencing skills. And it was the McGill was involved in the yeast genome project, and they were sequencing chromosome 1 and chromosome 16. So the whole yeast genome is 12 million bases, but the chromosome 1, which is the smallest chromosome, is only 230,000 bases. And so that's very small by today's. This is like a few minutes on a sequencer today. Back then, it took us, we were doing slab gels, where it was manual sequencing. It took us quite a long time. And we of course, published many papers and so forth. So after that, after those years at McGill, then I went to NCBI. And so I worked on GenBank, and so I was responsible for the database. I was a GenBank coordinator at NCBI, and I was there for 5 years, and initially started to work on ESTs, express sequence tags, but then I got seconded to work on managed GenBank. And GenBank is one of the 3 open nucleotide databases, with the one in Europe and then one in Japan. And apart from we had obviously many machine rooms at NCBI, as you can imagine. And but on my desk, I had a Unix box, a PC, and a Mac. So I had all 3 because I had some things I had to work on. I had to sort of switch machines for different kinds of tasks. And I had about 25 curators working with me on GenBank. And so this is a growth, and you've seen many. Maybe you've not seen them, but there are many of these growth curves that have been published over the years. And this is the 5 years that when I was at NCBI, that's the growth that I sort of supervised. And of course, we're starting to panic, and because the thing is just growing. And then a few years later, we had actually the 25-year anniversary of GenBank, and I was invited to do a keynote at that and show the growth of GenBank. And this is where my yellow period was in the past. And so where we panicked was it got a lot worse after that, and it's still getting worse today. So it hasn't changed. So interestingly, there's like 1,000 times more sequences being processed every day at GenBank, and it's still the same number of curators that there were back when I was there. And so that part has not changed. So after NCBI, I went to Vancouver, and so I was there for 10 years. I got recruited by Michael Hayden, who was also at the CMMT, the Center for Molecular Medicine and Therapeutics, which is attached to the Children's Hospital. And also he's the scientific director of the Canadian Genetic Disease Network, which is a network that sort of led to bioinformatics.ca workshops, which I'll talk just a little bit more about later. And then I also ended up starting the UBC Bioinformatics Center at the Michael Smith Labs on campus. During those years, I also co-edited with my colleague Andy Baxavanis 3 bioinformatics textbooks. And during, I would say, those Vancouver years were probably the sort of the most prolific bioinformatics years with respect to publishing and generating various projects. I was in charge of the Bioinformatics Core Facility at the CMMT and at the Canadian Genetic Disease Network. I was a consultant at Merck Frost for 5 years. I started the Bioinformatics Center, and I wrote the books I just mentioned. And I was also tenured at the medical genetics at the Michael Smith Laboratories, and also an adjunct at SFU. And so tomorrow, is it tomorrow? I think it's tomorrow. I'm giving another talk about the 25 years of CBW. And so that's a separate, I'm just showing you one slide, but I'm doing a 40-minute presentation on that. And in there, we've done something in the order of 169 workshops over the last 25 years, and on various topics, some of which are represented on this slide. During that time, in Vancouver, I also was involved in, I had PhD students and writing papers about gene finding models and prediction of RNA structure RNA structure that predicted that we could predict with doing experiments. And so that's another way of doing bioinformatics, is you predict some things and then you go to the lab to see if your predictions are valid, and develop some workflow tools, develop some gene finding tools, and so forth. Another database that we worked on while I was in Vancouver was the BIND database, which is a biomolecular interaction network database. And basically the concept here is you have proteins, but they interact. And so this is a database about who interacts with whom. And the way I explain bind is I use the IKEA data model. And so when you buy a piece of furniture from IKEA, you get two very important pieces of paper. One is your parts list, and that's like the human genome. So you know which genes you have, and you know which proteins you have. And then the second piece of paper, which is very important, is how the parts fit together. And this is what bind is. It's basically telling you this protein fits with there in this position at that time in development, at that time during the cell cycle, for example. And so it references the parts from the parts list, but it then tells you who interacts with whom. And so you can get some very complicated. So if you take all the proteins in the yeast genome, then you can get some pretty crazy, what we call hairballs of meshes. But then if you can focus in on areas related to the cell cycle or related to cell division or related to protein synthesis and so forth, then you can see which partners. And so what you'll find is, yeah, some proteins together, oh yeah, these three I knew were involved in translation, protein translation, but these other two that interact, I did not know they were involved in protein translation. So you now have all the pieces of things that fit together. And so that's been a really interesting project. And then when I was at the Michael Smith Lab, we also recruited all the other bioinformaticians on campus, and they were part of the center. But the first, Paul and Anna on the top right there, the people are recruited to the center itself. And these are pictures of my groups. My group over the years while I was at in Vancouver, and the bottom right there with the CMMT, that's when I was at the Children's Hospital. Those are the other faculty members at that center. And at the bottom right is a Van Bug picture. So that's a Vancouver Bioinformatics user group, which is a group of seminars basically that we presented that involved everybody from the Vancouver Bioinformatics community. And so we started Van Bug in 2002, I think, a year before this picture. And when I came later on, I was involved in Torbug and Monbug and so forth. So there are bugs all over the country. So after my years in Vancouver, I went to Toronto, not for the Maple Leafs. And so, but rather I came to Toronto to work at the Ontario Institute of Cancer Research. And Tom Hudson was the director of the OICR, and he recruited me, and I knew him from the Canadian Genetic Disease Network. And so he worked with him, and he was actually a supporter of the workshops as well. So it was quite interesting. And he was involved in a very interesting large-scale project, which was the ICGC, the International Cancer Genome Consortium. And just a quick overview of what the ICGC, basically building a database of about 50 different tumor types, and to get 500 tumor per tumor type. So that's 25,000 genomes from 50 different tumor types, because you have controls and normal tissue and cancerous tissue. And so it ended up being a project of 50,000 genome project. And it's an international project. And so you have people from all over the world involved. And this was a picture from the first meeting we had in Toronto. And so there's several papers that came out, actually on the order of about 50 or 60 papers that came out of that project. And I was involved with some of them more directly, and also papers from my own lab. So this sort of takes on the big data issue, sort of comes online, and you have to sort of give it some historical perspective. When I started in graduate school, or even before 1999, there was no human genome had been done yet. We did the first yeast genome The first C. elegans, the worm genome came out. The first Drosophila genome came out. The first bacteria genome came out while I was at NCBI. And so these are large genomes, but it's not big data yet. And so on the left-hand side here is a 5 meg hard drive in 1956. And that's basically an attachment on your email now. And on the right is a 5 terabyte. Or I have one, maybe it's even 2, 3, 4 times that on my desk. And so that's 1 million times more data size-wise, and that fits at your next to your laptop. The bioinformatics workshops have continued to flourish very much while I was in Toronto. And on the picture, in this picture on the left, top left here is a group picture of the whole bioinformatics team at the OICR. And there's about a 4-to-1 ratio of bioinformatics people to genomics people at the OICR. So that gives you an idea of the importance at this institute, but also the challenge that it's relatively easy to generate data. It's a lot harder to interpret it. And so that was some of the things we've done over the years. And I also want to highlight Michelle Brazas here on the middle right. And she, I recruited her from British Columbia, and she came to me to Toronto. But she also, I've since left Toronto because I'm in Montreal now. Michelle has taken over many of the things that I was involved in. And so she is doing a superb job. So important things to keep in mind is that open access, open data, and open source is essential for science. Openness is a responsibility for us to, and an obligation for something that if we are funded by the public, if we're funded by the public to do our work, then we have to make our things open, and because we have to give back to the public, basically. And so I'm now, after 10 years in Toronto, I'm back in Quebec. I did some consulting at University of Toronto for developing a professional master's program, bioinformatics master's program. I worked a couple years at Genome Quebec as their VP of Scientific Affairs. Also worked a year as a Montreal Neuro. And I'm sort of slowing down my career now. But I'm still very busy. I'm an editor for 2 open access journals, Database and PLOS Computational Biology. I'm a fellow of the society here. I'm a co-applicant with a team that just got funded last year to do a Canadian Human Genome Library, which is basically a database that will be held in Canada for genomes that are sequenced in Canada. And I'm the honorary chair of the meeting this year, and I'm not planning to move again. So he says. So in case you lost track of all my movements here. So if we start with 1 is where I was born in Quebec City. Then I went to Montreal. So I keep coming back to Montreal, basically, is the lesson here. So after Montreal, I went to Ottawa, then went to Winnipeg. Then I went back to Montreal. Then I went to Paris. Then I went back to Montreal. Then I went to Calgary. Then I went back to Montreal. Then I went to Vancouver, then back to Montreal. And then I went to Toronto. Then I went to Vancouver, Toronto, and then Washington, and then back to Montreal. So I'm here now. So what has driven me throughout my career? So to do biology. So I'm a biologist by training, I would say, and I've always wanted to do biology. But I do it using computational biology. I promote the practice of open science, and I support scientists. That's basically what I've done my whole career. Is I do that by publishing, I do that by teaching, by mentoring, by running bioinformatics core facilities, helping others do their bioinformatics, by being editor on open access journals, by supporting the funding enterprise, be it CIHR, Genome Canada, and so forth. And I served on many, I didn't mention that, but I served on many scientific advisory boards throughout all this, and also helping organize conferences. And so lessons learned. So what is it to do bioinformatics? I mean, I definitely highlight in my personal development the biological perspective, but there's lots of bioinformaticians that come from a CS background or a math background and have done great things. But this is from my perspective, and you can write software and document it. You can build biological databases. You can be a tool user and interpret genomes, human diseases, yeast phenotypes, plant growth, protein structures. And you read papers and create genome and pathway databases and curate these databases. And that's what some bioinformaticians do. All they do is read papers and build databases. And they're contributing to databases like Reactome, SGD, and so forth. You can run a core facility, or you can lead a group at your favorite university or a company. And so again, bioinformatics is about integrating biological themes together with the help of computer tools and biological databases and gaining new knowledge about the system in study. So some lessons I've learned along the way: do as I say, not as I do. Bioinformatics is an important science and part of the approaches, one of the approaches necessary to understand life. It's not the only one, but it complements the other ones very well. Personally, I've learned biology first, and then I learned to program, and I definitely recommend things like Python or R, and R, actually both of them are very good. And the thing that's coming up more and more is you have to learn a workflow language. That's sort of becoming necessary evil in our times. And not all things are done the same way. So people learn different things in different orders and so forth. Teamwork and networking is also important and essential. And this lonely scientist like Jean Rostand is not possible anymore. There's a paper, one of the papers we published in the pan-cancer analysis of whole genomes, we had about 700 co-authors in that paper. And so none of us could have done that paper by ourselves, and we needed all of those people to make that possible. I mentioned where I've lived, but I didn't mention all the travels I've done, and I've traveled quite a bit, and I definitely encourage you to travel for conferences and being on various boards or whatnot as much as you can. And definitely take care of your heart. I also did not mention throughout all this, I got married twice, got divorced twice, had another partner, she passed away, had 2 kids in there, and all of that is sort of part of life. And you have to take care of your heart and in the process. Don't forget that. And definitely good to read different books, and some of them you reread them again. So that's also a very useful tip. And most important is have fun. And then the last one is be open so people can see how great you are. And that's the end of my talk. Thank you. Merci. Any questions? Any questions? And I can answer questions in French if people want to ask me in French. No questions.
It doesn't look like there are any
Okay.
online.
No online. Okay. Anybody in the room? Okay. Thank you very much.